Hello guys, welcome to another video. In this video we're going to learn about the cloning vector. So let's get started. What is cloning vector? Cloning vectors are DNA molecules that are used to transport cloned sequence between biological hosts and the test tube. Here you can see the cloning vector, and now we're going to learn about the properties and features of vectors. Vectors must be able to replicate autonomously in the host cell and must have at least one usable restriction enzyme site for the introduction of foreign DNA. Cloning vectors share four common properties: ability to promote autonomous replication, contain genetic marker, unique restriction sites, and minimum amount of non-essential DNA to optimize cloning. Here you can see that is promoter, then restriction site, multiple cloning site, that contain genetic marker, restriction site, DNA, and autonomous replication. Here all the properties are included here. So then, what are the types of cloning vector? Cloning vector is mainly 6 types. First one is plasmids, phage lambda, plasmid phage hybrids, cosmids, BAC, and OAC. So in this video we are going to learn these types of cloning vector in briefly. So let's get started. First one is plasmids. Plasmids are circular double-stranded DNA molecules that exist in bacteria and the nuclei of some eukaryotic cells. Here you can see the plasmid. This is origin of replication, antibiotic resistance gene, selectable marker, restriction site, gene insertion, restriction site, and this is promoter. So this is the picture of a plasmid vector. Then important features of plasmid. Three features of the plasmid cloning vector, such as multiple cloning site. Here I can see that multiple cloning site. Then an origin of replication, where I can see the origin of replication. Then and gene specifying resistance to an antibiotic. This permits selective growth of the host cell. Most of often used resistance to ampicillin, penicillin, tetracycline, and kanamycin, etc. Then why plasmids are good cloning vector? We know that plasmids are good cloning vector. Why? Because they are small in size, easy to manipulate and isolate, circular, more stable, replication independent of host cell, several copies may be present, frequently have antibody resistance. Then along with these properties it has some disadvantages. These are can't accept large fragments, sizes ranges from 0 to 10 kb, and standard methods of transformation are inefficient. Then phage lambda. Phage lambda is bacterial virus or bacteriophage that infects the bacterial species Escherichia coli. It is discovered by Tusar in 1950. Here we can see the phage lambda. This is head and this is collar and tail. Then this is base plate and tail fibers. That is bacterial virus. Phage lambda is a bacterial virus, and this is the binding proteins. Then what are the properties of phage lambda? Phage lambda virion has a head which contains viral DNA genome, a tail which functions in infecting E. coli host cell. Then advantages over plasmids: they infect cells much more efficiently than plasmid cells. The yield of clones with vectors usually higher. Because of its widely used efficiency, phage lambda is often used in DNA library constructions. Then what are the advantages and disadvantages of phage lambda? Advantages: accepts large inserts, approximately 25 kb. Efficient infection compared to plasmids. Produce tiny plaques. They produce tiny plaques. Tiny plaques can screen. Then what are the disadvantages of it is not as easy as working with plasmids. Then cosmid vectors. Cosmid means cos sites plus plasmid equal to cosmid. A cosmid is a type of hybrid plasmid that contains a lambda phase cos sequence. Cos sequence have cohesive ends, always DNA packaging. They were first described by Collins and Horn in 1978. Plasmids are also a hybrid combination of phages and plasmids. Example, cosmid. Here you can see the cosmid vector, and this is the cos site. This is the cos site. Then what are the properties of cosmid vector? Package large inserts and thus are ideal for genomic libraries, and have a selectable marker, have an ORI origin, have polylinker with multiple cloning site. Here you can see that polylinker with a multiple cloning site. Then cos sequence then are packaged into defective phage virus particles, produces colonies. So these are the properties of cosmid vector. Then BAC, that is bacterial artificial chromosome. A bacterial artificial chromosome is a DNA construct based on functional fertility plasmid or F plasmid, that is fertility plasmid, used for transplanting and cloning in bacteria. Usually E. coli is used. BAC or bacterial artificial chromosomes were developed in 1992. BAC is useful in the sequencing of the human genome. Through the use of BAC, transgenic mice are developed. Here we can see the BAC, bacterial artificial chromosome, which is a cloning vector. Then what are the properties of BAC? BAC can hold up to 300 kbs. The F factor, that is fertility factor of E. coli, is capable of handling large segments of DNA. Recombinant BAC are introduced into E. coli by electroporation. Has a set of regulatory genes, ODS replication E, which control F factor replication, and par A and par B, which limit the number of copies to 1 or 2. A chloramphenicol resistance gene is and a cloning segment. Example, PBAC on 08L. Then OAC. OAC means yeast artificial chromosome. Yeast artificial chromosome are genetically engineered, derived from the yeast DNA, Saccharomyces species, which ligated into a bacterial plasmid. Here you can see the yeast artificial chromosome. This is the restriction site, restriction enzyme, then marker A, marker B, then this is origin, and this is cutting of cutting with restriction site, and sticky ends which can insert the foreign DNA. This is the linear form, and this is the circular form. So this is yeast artificial chromosome. Yeast artificial chromosome plasmid equal to yeast chromosome. Yeast artificial chromosome can hold up to 500 kb. OAC are designed to replicate as plasmids in bacteria. OAC contain a yeast centromere. Here you can see that is yeast centromere. Then 2 yeast telomeres. Here you can see 2 yeast telomeres. Then bacterial origin of replication and bacterial selectable markers. Then once a fragment is inserted, OAC are transferred to cells. They then replicate as eukaryotic chromosomes. DNA is inserted to a unique restriction site, and cleaves the plasmid with another restriction endonuclease that removes a fragment of DNA and causes the OAC to become linear. Once in the cell, the ROAC replicates as a chromosome, also replicating the foreign DNA. So this is all about the cloning vector, all about BAC, OAC and other plasmid, phase, lambda and other vectors. So that's all for today. See you in the next video. Thanks for watching.